Good afternoon. I want to thank everyone for joining us for our next installment of our Adventures in Field Research seminar series. We are very pleased today to have Dr. Joe Da Silva with us from Norfolk State University, and he's going to talk with us about present and give a presentation that he calls Parasites, Hosts, and Pursuits. I will remind everyone that there is a question and answer discussion period at the end. At that point, we'd encourage you to unmute your mics and start a discussion, and you can always use the chat option at the bottom and lodge your questions then. So with that, I'll turn it over to Joe.
Good afternoon, everybody. Thank you, Dr. Rollins, for giving me this opportunity to present to you. Thank you again, once again. It's a sunny afternoon from where I am, sitting in my house in Suffolk, Virginia. So this jaunt is about parasites, that is, in pursuit of parasites. And I want to begin by talking to you about how it started. It started when I opened my mouth. I had just finished my final undergraduate examination, and a Catholic priest professor asked me what I wanted to do next. I opened my mouth and I said, I want to study parasites. The next thing I know is I'm sitting on the back of his motor scooter, and we are headed for a government livestock research institute 10 miles away.
Hey, Joe,
Yes.
I can't see your screen. Are you sharing?
Oh, sorry.
Oh, no, you're fine. I just wanted to check and see if you were sharing.
I was. What happened now? Excuse me. Oh, wait a minute. Thank you. All right. Is that better now?
Yes, sir. I got it. Thank you so much.
All right. So as I was saying, that's how it started. We went to a government-run livestock research institute, and I met a man there who was a parasitologist, and he was a consultant to the government at that time. So he told me to go to his lab the next morning at 7 o'clock. And he also told me to tell my folks at home that I wouldn't know what time I'd be going back home. And so at 7 o'clock the next morning, I find myself with the intestine of a cow. It's 132 feet long. And he gives me a scissors. Then he says, open it up. And then the next thing I know is I have buckets and buckets of feces that I'm sifting through and looking for parasites. So that was that. I trained as an apprentice, no money, no pay, for as long as I could. Then when I went to the University of Dhaka to study for my master's, my professor there put me on to study parasites of fishes. So this is what it is all about. It's about pursuit of parasites, and pursuit of parasites in fish particularly. This is where my adventure all began. John, how do I get to the next slide? Okay, there it is. Good. Now, this is showing you where Bangladesh is. Bangladesh is located to the east of India. On the top of us, way up on the top is Nepal and Mount Everest. And to the south of us is where the Bay of Bengal is. And that is where I was doing my sampling. Here is a map of Bangladesh to show you the area that is the Bay of Bengal. It is to the south, and you can see it in dark there. And then where my pointer is you can see a city called Cox's Bazar. That was my sampling site. I also wanted you to see this map to show you the rivers that we have. These are very, very large rivers. They will come up in our discussion a little later. This is to show you, this is to show you an area in Bangladesh, in the Bay of Bengal, where I did my sampling. This area has been blown up a bit. Now, I was there to collect parasites from fish and to identify them. Taxonomy of parasites. The parasites that I collected belong to the phylum Platyhelminthes. So these are flatworms, and they are trematodes, and they are digynians. Digynians have a two-host life cycle. Digynian parasites do. One of the hosts is a vertebrate animal, and the other one is an invertebrate. And the invertebrate is always a snail. So digynians have a snail host in their life cycle. The character by which we identify a platyhelminth trematode parasite is by looking for two suckers. These suckers are an oral sucker and a ventral sucker. So I want to begin by talking to you about fish as hosts of parasites. And this is part one. You can see two of the fish that I opened up to look for these parasites, but we will see some more fish over here. Here are the two you saw earlier on. One of them is known as a cleft belly. The other one is known as a wolf herring. And the other one is known as Hilsa shad. I want you to remember this fish particularly because it is going to figure in our discussions later on. So its scientific name is Hilsa ilisha. You can see it in the box there. And the shad is very similar. The Hilsa shad is very, very similar to the fish that you have in the United States because it's a migratory fish. It migrates from the salt water to the fresh water. So you can see three of the host fish that I opened up to sample for parasites. Over here on the third column, you can see the names of the parasites that I was able to collect from these fish. Here are two more pictures of the host fish, Hilsa shad and a hairtail. Now this Hilsa shad that you see here is a bit different from the one that I showed you earlier. And again, on the third column that we have over here, I show you the names of the parasites that I collected. Two more pictures to show you the fish. One is a hairtail and the other one is the Indian mackerel. These are three more host fish. One is the yellow striped scad. The other one is the pomfret. And the 10th one is also a pomfret. The one on the top is the black pomfret and the one at the bottom is the silver pomfret. And then you can see the names of the parasites that I collected from these fish as well. Commercial break, time for commercial break over here. I need to drink some water. No, I was just kidding. This is to show you conditions in Bangladesh. It's rainy when it's the monsoon season. The rain just comes down on you for at least 3 months. And then it's hot and it's muggy and you sweat and you perspire. So not the ideal conditions under which to go looking for parasites in fish or to collect fish for parasites. But heck, who said that ecology wasn't fun? There's no end to surprises, in my opinion. This is a slide to show you these platyhelminthes or trematode parasites that I picked up. And one thing you can notice very, very definitely are the two suckers. Here's the oral sucker, and here is the ventral sucker that I was talking to you about. And then if you can see the print over here, the labels that we have over here, these are the structures that we look at when we try to identify them. There's the vitellaria there, that's the pharynx over there, that's the mouth over there, and then there's the reproductive parts over here, VS, CRS, PP, PC, and so on. Now, notice very, very carefully that this is where we have two testes. And then this is where we also have, where my arrow is, where we have the ovary. But a lot of the parasite is filled up with the uterus that contains a lot of eggs. Here is the uterus showing you a lot of eggs over here. This is vitellaria over here on the sides. So all of these characters are used for us to identify these parasites. This is an egg that you can see over here, and so on. And here is a blown up part of the parasite to show you the oral sucker, and then to show you the ventral sucker. These are two more parasites, trematodes, and you can see that they are quite different from the one that I showed you in the last slide. This one is kind of short, but it's also very, very broad in the middle around here. And the sucker is very, very prominent. You can also see the uterus, and you can see the vitellaria over here on the two, sorry, this keeps, it's very sensitive pad that I have over here. So getting back to showing you the structures that we use to identify these parasites, with the vitellaria here, of course, oops, we have the vitellaria, and then we have the uterus over here, and then we have the ovary over here. And you can see in this one, in the case of this one here, that the uterus is filled up full of eggs. So these parasites, they shed a lot of eggs in the host fish that they live in. Another slide here to show you a different species of trematode. This is Lacetocladium magna acetabulum, because the acetabulum or the ventral sucker is so large. The acetabulum is another name for the ventral sucker. And you want to notice over here that this part here is the body of the parasite. This is known as the soma. The lower part, that is this part here, is the tail, or it's known as the exoma. So we have a soma and we have an exoma. And in this case, in the case of this Lacetocladium magna acetabulum, you can see that the uterus filled with eggs has descended into the exoma. So it's quite a robust worm in the sense that it's quite stout, quite short, but it contains a lot of vitellaria. Sorry, it contains a lot of uterus with eggs in them. And these are the two testes that are very, very prominent in this case. Lacetocladium dawesi, as you can see, is a long worm, and it's got a soma and it's got an exoma. The uterus descends into the tail of the parasite. This is to show you some more parasites. Okay, this is to show you over here another Lacetocladium species, and you see how it is short, it's stout. Here are the two testes over here, the ovaries over here, and then we have the uterus again filled up with eggs. But the exoma is not all that well developed in this case. And you can see over here details of the anterior part of the parasite to show you the oral sucker and the other structures that we use to identify these parasites. This is another one over here. Notice it's taller than the one on the left-hand side. And this has got exoma as well, but it's not all that well developed. Notice the position of the uterus over here. It's way up on the top over here, but below the testes over here. This table here shows you the location of the parasites in the organs that we found them in. This is a list of 14 of these parasites that I was able to identify. And this gives you over here the organs. You can see highlighted in yellow is the stomach. So the stomach doesn't contain all that number of parasites, but the intestine does. The intestine seems to be the favorite organ for these parasites to occupy. But that is what is to be expected because the intestine is where most of the food is. Looking at the rate of infection, we examined as many as 100 fish. When I say we, I mean I had a peer of mine, a classmate of mine. We went looking at these fish for parasites. He looked at the parasite that occur in the gills of the fish. And I looked at the stomach and the intestine. So I found out that 52.63% of the fish were infected. Now, if you look at the numbers of species of parasites infecting each one of these fish, you will see that the Hilsa ilisha, this is the migratory fish that goes up the rivers to spawn, had the most number of species of parasites, 4 species. It was followed by this other one known as Celeroides leptolepis. It had 3 species. And then the rest of the 10 fish that we looked at had only 1 species of parasites in them. Good. Now I want to come to the second part of the, before I start with the second part of the Hilsa as Parasites of Tags, I want you to understand that sampling for parasites in a place like Bangladesh is really an adventure because it's an underdeveloped part of the world. And in the time that I was talking about, we only had these buses that we could travel by from one place to another. And it was an overnight trip. And then when we got to the place where we were doing all this study, there wasn't a table, there wasn't a chair, there wasn't any lab space for us to do all our dissections. There was an open place where the fishermen brought in their fish, and so we had to sample fish from there. Part 2, Hilsa parasites as tags. So you have to fast forward 22 years of my life when I started looking at this one particular host fish, which was the hilsa. And the hilsa, we know, had a number of these parasites. The hilsa is a migratory fish. It migrates from the Bay of Bengal into the estuary, brackish water, and then goes into the fresh water to spawn, to lay its eggs. So we were very interested to see if we could use parasites as tags to see where the fish went to lay their eggs. That is where the fish went up the stream in the rivers in Bangladesh to lay their eggs. And this would be a big help for people who were doing population studies of hilsa. Now, I use the word we over here because I had a number of colleagues join me in this research effort. They were from Norway. And then, of course, I had my students doing their master's degree or doing their PhD degree. And so we had quite a big team with us. And you can see a student of mine and myself. The photograph is not very, very good. It's a black and white photograph. And we are in one of the largest mangrove forests as well as a jungle in Bangladesh. This is the Sundarbans, the home of the Royal Bengal tiger. Now, this may not mean anything to a lot of you, but you don't want to be around when those tigers are there. They growl and you can hear them, but you'll never see them. But they can see you, and you don't want to be around when they are around also, because you can be dead meat. You can be dead meat. This is a map of, this is a map of Bangladesh again to show you the rivers that we have there. You can see that they spread out like arteries and veins in a person, in a human being. And of course, the fish live in the Bay of Bengal, this hilsa shad, and it goes through the brackish waters over here and goes up this way, goes up this way, goes up this way, goes up this way, every which way that you can think about. So it was up to us to try and now sample fish from all these places, from all these areas, and then find out what the parasite content was for these fish, of these fish. See, when I talk about it like that, it seems so very, very easy. But when you are there, it becomes an adventure. It becomes an adventure because, first of all, you don't know when the run is going to begin, when the hilsa fish run is going to start. We know, rule of thumb, you know, Benjamin's Farmer's Almanac, that it's going to rain in the month of May. The monsoon rains are going to come down on us. And as soon as the first drops of the monsoon rain hit the water, the hilsa fish, hilsa shad, is going to make its run to lay its eggs. So until then, you never know. And then, of course, when it starts to rain, you start traveling. Again, you travel by bus, you travel by boat, and you go to these remote areas, and then you don't catch the fish yourself. Fishermen do it for you, and then they bring it to you, and then you open them up, and then, of course, you take the intestine and the stomach and preserve it and bring it back to the lab, and then you start to do your sampling. So those were the conditions under which we were working. Now, there was this one time when the fisheries officer, I've got to tell you this, when the fisheries officer from this area here phoned me and said to me, to get there right away because it had started raining and the fishermen were catching fish. So we took an overnight bus, landed there in the morning, and then hired a boat, and then went down to the place where these fish were spotted. And there were fishermen fishing there. And then guess what? After waiting there for about 12 hours, we were only able to sample one fish. One fish. And that trip cost me a lot of money in Bangladeshi monetary units. So that's ecology for you. Well, here is ecology for you because this shows you the sites where we did our sampling. The places were freshwater, estuary, and seawater. We sampled 4 freshwater sites, and these names won't mean anything to you, but they are located far away from each other. They're nowhere near each other. And then the estuary, if you look at the names over there, they won't mean anything to you either, but they are strung along the south of Bangladesh. Cox's Bazar is on the southeastern part of Bangladesh. The parentheses, the numbers in parentheses tell you how many fish we sampled each time from these places. Sometimes we couldn't sample as many fish as we would have liked to. For example, in Sherpur, we got only 11 fish. We were able to sample only 11 fish. In this other place in the estuary in Patuakhali, we were only able to sample 21 fish. So that's that. We travel to all of these places, and that's where it is an adventure because you never know what's going to happen until you get there. Is it going to be raining? Is it going to be hot? Is it going to be humid? And where are you going to stay the night? Where are you going to eat? It's not like a place in the United States, okay? This table here shows you all the parasites that we were able to identify from the Hilsa shad, when we were looking for migratory routes. So you can see one of my earlier parasites that I identified, Fostula species, was the most prevalent parasite in the Hilsa. And it was quite abundant too, in the sense that up to 40% of the parasites were Fostula. It was followed by another hemiurid or platyhelminthes parasite, which is Aphanorus tosici, and prevalence was quite high. 75% of the host fish Hilsa had parasites, excuse me. The abundance was also not bad, quite high, excuse me. But you look at all the other parasites that we collected from here. While the prevalence was quite high, the abundance was low. I also want you to understand that all of the parasites that you see on this list are not trematodes. Some of them are cestodes, some of them are also nematodes. And they are not all that great in numbers, as you can see. For example, you can see C. llewellynii, 20% prevalence and 3% abundance. And then you can see another cestode parasite there. The rest of them are cestodes. And then again, we have nematodes over there, Capillaria porosicum and Camelinus. So that is that. This shows you the distribution of these parasites in fresh water, in brackish water, and in marine water. The topmost figure here shows you distribution in freshwater, the middle one in brackish water, and then the last one over here in marine water. We are also looking at the organs that these parasites occupy. And look very, very carefully, you will see over here, if you can see my pointer, that this is the cardiac portion of the stomach. This is the cardiac portion of the stomach, and this is the cardiac portion of the stomach. So the cardiac portion of the stomach harbors quite a lot of parasites, but nowhere near the number that we find in the intestine. The intestine is the place where most of these parasites are found. Interestingly, the stomach itself doesn't have a lot of parasites, interestingly. And the picture is the same. The picture is the same in fresh water, in brackish water, as well as in marine water. So the picture doesn't change about the location of these parasites, whether the fish is in fresh water or in brackish water or in salty water. Now, what is it that we can conclude from this? We concluded that we cannot really use parasites to tag this migratory fish, Hilsa, when it goes up the rivers. It seems like, it appears that the host fish, Hilsa, can harbor all these parasites in the same organs throughout the change in the environment. That is going from a salty water situation to a freshwater situation. You would think that the fish would be able to do that because it's migrating physiologically. The water, the tonicity of the water should have an effect also on the parasites, but it does not seem to have that effect. So that is what we concluded as a result of this study. I will end my jaunt over here. And before I open my mouth anymore, I will take your questions. But after your questions, I would like to speak again, if I may.
All right, I'll ask a question. I'm curious. You mentioned being able to hear the growling of the tigers. Did you have any close encounters with tigers in your field work?
Did I? I'm smiling because, as you can see the slide over here, this was in the jungles over there. And we did hear a lot of the tigers, and we did see pug marks on the ground, fresh pug marks on the ground. So that meant that they were there somewhere. And interestingly, when we went to do any sampling to this part of Bangladesh, which is the Sundarbans, we had to live off a boat. We were not allowed to live on land, even though there were places we could have lived over there. And the reason was that the government didn't want us to become prey to these tigers. That's the reason. So we could hear tigers howling all night. One night we went up to a big perch that they had built for us to see out in the distance. And we waited all night for a tiger to show up, and it did not. But to answer your question even more, people get killed. People who go into the forest to cut wood to bring back to land to be used as firewood. And then people go in there to harvest beehives for honey. And so they get killed as well. So does that answer your question?
Yes, that's dangerous, and tigers must not be very good at swimming, so stay on the boat.
Right.
I have another question. You have done this work with parasites, and these are parasites in fish. While you and your crew are doing this field work, did you ever have any problems with any types of parasites that would mess with you?
No, fish parasites that way are quite good. Quite good. But I studied another parasite for my PhD work, and this was London University in England. And the parasite I was working with was that of a rat. It lived in the rat, and it's a pinworm parasite. It comes out during the daytime and not at night. So it has a diurnal pattern of appearance. And I wanted to see whether it would do the same thing if I took some eggs, ate it. And so I did that. I infected myself. It's not very nice. It's not very pleasant to hear. But believe you me, it adapted to my intestine. It adapted. So that's that. The parasites that you have to be careful if you go into the tropics is always these protozoan parasites, Intermoeba histolytica, that can cause amoebiasis or giardiasis. Those are protozoan parasites.
Okay, Joe, so that's awesome. Like that is, I know how dedicated you are.
Thank you.
I like fishing, and I was just wondering, when you're doing sampling, is it traditional? Like, is it fishing, or is it like throwing out nets? What are you doing when you're obtaining a sample?
Now, the fish that I obtained, that I dissected, were brought in by fishermen,
Okay.
and they were using nets.
Okay.
They were using nets.
Okay.
Very fresh,
cool. Awesome.
Very fresh fish.
And then this is a shock factor thing. So when I think of parasites, I always have horror stories, flashbacks back to high school in biology with Martha Gunpool. I can even say the teacher's name. Showing images of 4 or 5 foot long tapeworms, those type. Have you ever seen anything like that? What's the largest parasite you've seen?
Tapeworm. The tapeworm that lives in pigs, for example.
Mm-hmm.
Tapeworms always live in the intestine of animals. So that one can grow to be 1 meter long. Huh? Mm-hmm. They're quite big. And some of the other parasites, roundworms. Oh, I don't know if you can see my ruler over here. They can be as large as 15 inches. It's not uncommon. It's not uncommon. If you go to the tropics, any child, any person there will have roundworms, will have flatworms in them. These tapeworms, especially, they'll have them. Pinworms as well.
Thank you. I'm more terrified to go swimming in waters now than I was before. So I appreciate it.
Listen, in the United States, that danger is not there anymore, except for one protozoan parasite. Which can get into your brain. But that is also very, very rare.
Is it Naegleri fowleri?
No, I don't think so. Naegleri, no. No, that's not it. That's not the one.
I had one question.
Yes.
So I know you said that you worked with the local fishermen and they would bring you their catches. How do you start that relationship with those local fishermen? And I guess, is that something they're used to with researchers in the area?
They are not. For them, it's a novelty. But people are very, very friendly. Fishermen are, you approach them and then tell them, you talk to them and then say, look, you want to do this and that. And then, okay, they get interested. And so they'll give you their fish. Of course, we pay them for it because it's their livelihood, right?
Mm-hmm.
Now, some of these fishermen go out on overnight trips to catch their fish in the Bay of Bengal. And some of them go out there for days on end, maybe 10 days.
Wow.
And then they come back and then they sell their fish. So that's that. I'm glad you asked me that, because we take it for granted, that, okay, they're there, we'll go to them and they'll let us have some of their fish. Yeah.
Yeah, I think that's some of the, I guess, the little things that people don't necessarily think of when we say, well, we have all these fish, and like John, his question of where do you get the fish, and then just thinking of going out there and seeing the local fishermen and starting that. I know it's just something that people may, like you said, take for granted.
Yeah. With the people who go out and catch fish, catch hilsa, that's a very, very big part of their livelihood. And as I said to you before, the conditions are really primitive compared to life in the United States. And a lot of these fishermen lose their lives going out fishing because all of a sudden a storm will whip up in the Bay of Bengal, and that's it. Their boats overturn and then they lose everything. So that's that.
Can you tell us a little bit, give us some insight as to why would we want to study parasites?
They're exciting. They're very exciting because they're so teeny weeny, nobody wants to look at them. But at one time, they played a very, very important role in the lives of people because so many people got infected, died, etc. And then you have to take into context here the history of the world. The British, you see, had their colonies everywhere, and they had soldiers in these countries. That is, they had soldiers in these countries from the 1700s, 1800s, and they have to look after their soldiers' lives. So they started all these research institutes to look into parasites, and they pumped a lot of money into that. Not only they, but also the pharmaceutical companies pumped a lot of money to come up with drugs. And as soon as the British lost their colonies, we also lost research funding, and then we also lost the support of these drug companies. But the interesting part of parasites is they're so small, they're so tiny, and they do a lot of damage to a lot of people. For example, sleeping sickness in Africa. For example, bilharzia in Africa again, and then malaria. I've sampled so many smears, blood smears, that I can't tell. Looking for these teeny weeny little parasites that will only get into the red blood cell. They're still dangerous, still dangerous. But if you don't have any more questions, then can I say something more?
Yes, please.
I mean, this was about adventures in ecology. And I have had more than one adventure in ecology. And that's the reason that I wanted to speak today. For example, my friend and I, we have been shot at sampling fish for parasites. And this was in British Columbia in Canada. So we went out on an overnight trip. We laid out our gill nets in this lake. And then the next morning we went to pick up our gill nets. And then people who were living around the lake started taking pot shots at us. And then, of course, we continued our sampling. And then sooner than later, we see the speedboat coming at us in full speed, comes up to us, stops us and says, "What the hell do you think you're doing?" And he's the ranger. He's the local fisherman ranger, fisheries officer. And it's a Saturday morning. And so he is even madder than you can expect them to be. And then he says, "Look, this is not the fishing season and you cannot fish. And therefore I'm going to arrest you." And at this point, of course, my classmate, my friend, he pulled out this permit he had to go fishing. And then, of course, the officer gets even madder. He says, why didn't you tell me this in the first place? If he had been told in the first place, he wouldn't have had to come out to catch us there. And that's when my friend told him that he had telephoned him several times and he never picked up the phone. So that was one escape that we had. And the other one was, again, collecting parasites in frogs. And my friend and I, this is another friend of mine, and we went out to a lake again. This is in British Columbia, Nanaimo. It's an island over there, Vancouver Island. We went out there and there was land in the middle of this lake. And so there was a pine tree that had fallen on the lake itself, and it was bridging the land and this little island. So I walked across this. It wasn't so difficult because I'm used to it, living in Bangladesh. And then I sampled over there. And then the next thing I want to do is get back on land, except this time I don't have the courage to go across this pine tree. And so guess what I did? I started to swim, waders and all. And that's when I nearly drowned. So twice in my life, I've gone out doing work and then I've nearly lost my life. And of course, my friend at this time threw me a lifeline. He threw me a rope and then he pulled me across. But I've had all kinds of adventures. And that's why I was attracted to this proposition that you had to speak. Thank you very much for listening to me.
Absolutely. And we want to extend a great appreciation and thanks to you for sharing with us some information about the work you've done. And you certainly have had some adventures. There's no doubt about it. With that, I think we're going to, we'll give one more chance for questions if there's any additional questions. All right, hearing none, I want to thank you once again. Very much enjoyed the presentation. I do want to express some appreciation to Lainey and John specifically.
Thank you, Lainey and John.
For working through and making this seminar series possible, handling all of the scheduling and logistical considerations. We certainly appreciate their work and contribution. And I do want to put in a bit of a plug for next week. I want you all to invite your friends, everyone here, email out, and use social media or whatever to invite a friend or two, or invite a class. Anyone can attend. We'll have a talk next week. We'll have Dr. Rachel Goodman from Hampton Sydney College, who will be talking with us about some of her adventures. Also encourage you all to check out Lincoln Memorial University and the School of Mathematics and Sciences on social media at Facebook and our Instagram accounts. So thank you once again, Joe, and I hope everyone has a great evening and a great week. Have some adventures and come back with a friend and see us next week. Thank you.
Thank you again. Bye now. Bye, John. Bye, Lainey.
Thank you. Have a great
Bye,
day.
Joe.
Thank
Have
you.
a good one.
Thank you much.
All right. Goodbye.
Bye.